This is Pushpalata from Vishaka Government Degree College for Women, Visakhapatnam. This video gives an overview of DNA replication. What is DNA replication and where does this process takes place? DNA replication is nothing but duplication of DNA molecule. This process takes place in S phase of interphase. We know that cell cycle has two main phases, interphase and division phase. Before division, that is in interphase, there are three subphases, namely G1, S and G2. In S phase, the actual process of DNA replication occurs. How the molecule of DNA doubles? There are the three models to explain the process of DNA replication. Conservative, dispersive and semi-conservative modes are the three different models proposed to describe DNA replication. These are the three different modes of DNA replication. The first type, that is semi-conservative mode. In this type, parental molecule gets doubled. The two strands of the DNA molecule, parent molecule, separates during the process and each strand acts as a template to synthesize the new strand. That means in the newly synthesized or in the new DNA molecule, there is an old strand and a newly synthesized strand. The old strand acts as a template for DNA synthesis. In conservative mode, the parent molecule conserves as it is and the DNA molecule, new DNA molecule is formed from the old molecule. In dispersive mode, the two strands are fragmented. Newly synthesized strand consists of fragments of old and new DNA pieces. These old DNA pieces are interspersed between the newly synthesized pieces. Here are the enzymes and proteins involved in DNA replication process. DNA ligase, DNA polymerase, nuclease, DNA helicase, DNA primase, DNA topoisomerase are the different enzymes involved and required for DNA replication. Single strand binding proteins are one of the proteins very important for process of DNA duplication. Steps of DNA replication. Formation of replication fork. Initiation and elongation of the chain. Termination of the DNA replication process. First step is the formation of replication fork. This picture shows how the DNA molecule forms new strands. First of all, unwinding of the double-stranded DNA takes place. The unwinding starts at AT-rich regions, adenine, thymine, nitrogen base rich regions. These are named as Origin of replication. These are the sites of origin. In short, ORI, origin of replication initiation. Origins are targeted by initiator proteins which go on to recruit more proteins that can help the replication process by forming a replication fork around the initiation region. ORI. DNA helicase, the enzyme which starts the unwinding of the DNA molecule from its ORI and exposes the two strands and resembling a Y structure referred to as replication fork. Helicase causes topological stress to the unwinded strand forming supercoiled DNA. While opening or while unwinding the DNA double strand, it causes supercoiled DNA. This stress is relieved by another enzyme called topoisomerase by creating negative supercoils in the molecule. The replication fork is bidirectional as the DNA strands are anti-parallel to each other. One strand is oriented in 3' to 5' direction and the other strand is oriented in 5' to 3' direction. But always the newly forming nucleotide strand progresses, the synthesis progresses only in 5' to 3' direction. The strand, that is 3' to 5' directionally the oriented strand, synthesizes the new strand in a continuous process, continuous manner. This strand is named as leading strand and the complementary strand on which the DNA synthesis takes place in a fragmented manner is known as lagging strand. Step 2. Initiation of DNA replication. One strand runs from 3' to 5' direction towards the replication fork and is referred to as leading strand. On this, DNA replication occurs in a continuous process. To this exposed single-stranded DNA, single-strand binding proteins adhere to prevent recoiling of DNA molecule and to stabilize it. Otherwise, the separated DNA strands will come to the original position to make double-stranded as usual. DNA primase comes into the action to synthesize a short stretch of RNA primer, which provides a 3' 3' hydroxyl group for DNA polymerase and can now add nucleotides and extend the new chain of nucleotides. And then elongation. Primer is added to unzipped two single-stranded DNA. These strands now act as a template for synthesizing new DNA molecules. The enzyme DNA polymerase synthesizes new nucleotide to match the template and add on to the free 3' hydroxyl group provided by the primer in each single-stranded DNA. The leading strand runs from 3' to 5', so the addition of nucleotides by DNA polymerase happens from 5' to 3' direction as the replication fork progresses The addition of nucleotide is continuous, thus only requiring the primer once. This is the picture showing the orientation of DNA polymerase enzyme, and it sits on the DNA where the replication initiates. However, lagging strands are anti-parallel and run from 5' to 3', and the continuous addition of nucleotides is not possible as the replication fork progresses. DNA polymerase cannot add complementary nucleotides to 5' end. Therefore, multiple primers are required to synthesize DNA on this lagging strand. That is why the process is discontinuous on lagging strand. Due to this phenomenon, the nucleotides synthesis from lagging strands occur in a fragmented manner. These fragments are termed as Okazaki fragments. Hence, the leading strand using only one primer synthesizes nucleotides continuously, while the lagging strand uses multiple primers and thus synthesizes nucleotides in a discontinuous manner. And finally, termination step. RNA primers of both leading and lagging strands are cut off or degraded by nucleases. The activity of polymerase and the nicks or the gaps so formed are filled with DNA and sealed by the enzyme DNA ligase. DNA polymerase also shows proofreading activity and check, remove and replace any errors. Interestingly, in eukaryotic organisms having linear DNA, when RNA primer at 5' end of daughter strand is removed, there is not a preceding 3' OH such that DNA polymerase can use it to replace with DNA. For solving this end replication problem, researchers have found that linear ends of DNA called telomere, these are used which contain specific GC-rich repeats. These sequences are known as telomere sequences. These telomere sequences do not code anything but are essential to fill in the gap in the daughter strand and maintain the integrity of DNA. Eventually, the replication fork terminates at terminating recognizing sequences, TER sites. The TER sequences are of around 23 base pairs which facilitate as the binding sites for TUS proteins. This TUS complex arrests replication fork and terminates DNA replication. This is the picture showing the entire process of DNA replication. The dark bands on the two strands are the initiator regions. In this initiating region, replication bubble forms. The attack of the enzymes takes place here, and progress of the enzyme DNA nuclease in 5' to 3' direction. After removing the primer, sealing takes place. Finally, after initiation and elongation, termination occurs. With this, the DNA replication process completes.